you. So today I'm going to be talking about micronutrient trace metal dynamics in the Western Arctic Ocean, which is one of my recent funded projects. But before I start, I just want to thank IOPSO and my nominators for this great award. It was a big surprise, and as Thomas said, a real joy in this kind of awful last year. So thank you very much. So in my talk about micronutrients in the Western Arctic, I really owe a big thanks to newly minted Dr. Laramie Jensen, who is the first PhD student to graduate from my lab and really led the analytical effort on this project. I'll start with Arctic Ocean climate change, which is probably no surprise to this audience, but it's disproportionately affecting polar regions of our Earth, which are suffering twice the global temperature rise since 1900 as the rest of the globe. And in the Arctic specifically, increased sea ice melt and river discharge have really increased the inventory of fresh water in the surface Arctic, which already received quite a lot of fresh water. Actually, it already received 10% of global river discharge to only 1% of total ocean volume. So the Arctic is a particularly fresh ocean basin and becoming fresher yet. Importantly for my work in chemical oceanography, these changes to the Arctic Ocean are not just physical changes, but are also biogeochemical changes with influences through chemistry and physics through the food web. As an example of that, I want to show you guys an annual temporal distribution of chlorophyll concentrations near Greenland in the Arctic, with chlorophyll on the y-axis and years are here in color. And Arctic primary production has this single peak. It's always had that. It's known to be limited by light over most of the year, except for here in spring when you get light and you get this huge bloom. And that bloom eventually breaks down due to nitrate limitation, or so we think, because Arctic biogeochemistry, as I said, is changing due to climate change. So you can see here that 2019 and 2007 were uniquely high production years, and trying to sort out why we have variation in carbon production in different parts of the Arctic is an outstanding question. Some hypotheses are that reduced sea ice coverage will result in more open waters that can support phytoplankton blooms, and also that rivers and sea ice supply essential nutrients and organic carbon, in that case, to the Arctic. And so we're doing a lot of thinking about how much the nutrient inventory of the Arctic is changing and where that effect might occur. So what's cool about this map here is changes in this 2019 peak year compared to the average of the prior 2 decades. You can see some places have had large increases in primary production, and other places have decreases. So my big question for the project, but also in a just bigger oceanographic question for the Arctic, is how do nutrients cycle in the Arctic, and how will climate change play a role in changing that and their role in the ecosystem? So my specific science has to do with trace metals, which have nano to pico scale concentrations, very low abundances, but these metals are essential micronutrients for phytoplankton in the ocean. So in this talk, I'll focus on manganese, iron, nickel, copper, and zinc, which together are involved in a huge number of cellular processes, ranging from photosynthesis to carbon and nitrogen transformations and cellular protection. And these metals are essential because they're used in the active sites of cellular proteins. So you can see this is a heme protein, and each of these green areas is an iron. And these phytoplankton that are growing across our global ocean have cellular requirement for many different nutrients, which here's the Redfield ratio that we think of for the macronutrients, carbon, nitrogen, and phosphorus, but also they require many trace metals, which we call micronutrients, in about 1,000-fold lower abundance. But these are still critically important because they also have, about 1,000-fold lower concentration. So equal to the micronutrients can be limiting to primary production. My Arctic project was part of the Arctic Geotraces program, which is an international program that's seeking to identify the processes and quantify the fluxes that control the distributions of key trace elements in the ocean, and establish the sensitivity of those distributions to changing environment. So this was the original set of spaghetti lines that Geotraces hoped to occupy across the global ocean. Yellow is what has been accomplished to date. And this is the US GNO1 Geotraces cruise, which took place in late 2015, which was, it turns out, the first semester of my professorship at Texas A&M, very busy time in my life. And we were excited to study this transect because the Arctic Ocean has uniquely large, broad, shallow continental shelves. About 50% of the Arctic by area is continental shelf. It also has those major rivers that I talked about. It's semi-enclosed, so it has known inputs and outputs from the Atlantic and the Pacific, and generally low primary production that separate it from the rest of the global ocean. And for the rest of this talk, I'll show my data in sections that goes northbound on the left from the shelf to the North Pole, and goes southbound on the right from the North Pole back to the shelf. So again, my driving question is, what processes control trace metal distributions in the Western Arctic? And prior to Geotraces, this is what we were working with. First, I want to draw your attention to these dates on these papers. Many of the papers on metals in the Arctic were from 30 to 40 years ago, when we had a lot of issues with our metal measurements, a lot of contamination problems, and the sampling was very opportunistic, just a site here or there, even with quite limited depth of the profiles. Right prior to Geotraces, some universities in Alaska were starting to get some more studies together of the Arctic, but again, they were very close to the shelf, kind of in their backyard. And so we really had a very little understanding of the Arctic micronutrient distributions prior to Geotraces. What we did have was some hypotheses, though, from that prior work, that rivers and shelf processes could be especially important in the Arctic, which may not be a huge surprise, but certainly was not proven by these early data. The first set of results I want to show that really, on a fundamental scale, blew our mind was that the Arctic has very unique profile shapes. So we had, these depth profiles with 24 to 36 points per depth, which is the most of any Arctic profiles of trace metals to date. And we had, much improved analytical methods over the last 3 decades. And so you can see here in the North Pacific data, I've got phosphate in gray, zinc in blue, which is a well-known and highly essential micronutrient, and silica in orange, in both the North Pacific and the North Atlantic. And these profiles have a characteristic shape showing biological uptake in the surface ocean and regeneration of sinking dead phytoplankton in the deep sea, causing higher concentrations to be regenerated back. And you can see the North Pacific and North Atlantic showed the characteristic Pacific concentrations being greater than the Atlantic concentrations due to the older age Pacific waters. So again, low in the surface, elevated at depth. And then this was our Arctic data. Looked really different, right? Kind of low still in the surface, huge peak, and then really low again at depth. So a couple of questions came up pretty quickly. Very low deep water concentrations. Where was our biological regeneration signal? And this peak, where was this peak in all of our nutrients actually coming from? Right in the subsurface, about 200 meters depth. So the first thing that we spent a lot of time thinking about was that peak, and it turned out that the highest concentrations correlated with a water mass called the Arctic halocline. This halocline forms when sea ice is formed over the Arctic shelves. You get brine rejection during that process, which sinks to the bottom and forms this denser salty water layer called the halocline. You can see this really clearly in our data. This is the Chukchi Shelf, and this is the North Pole. And this green layer with a salinity between 31 and 33 is the upper halocline layer. These are Pacific-derived waters that are influenced by sea ice formation over the Chukchi Shelf. And what was cool about this, again, you can see this in our salinity. And again, here's the Chukchi Shelf going north to the North Pole here in the middle, and then coming back southward again to the Chukchi Shelf in this V transect. So the upper halocline layer was this green layer, sort of between 50 meters and 150 to 200 meters or so. And we had, coincident with that upper halocline layer, maxima in silicate, maxima in dissolved zinc, maxima even in iron, which was a lower abundance, but still a relative maxima in our profiles in this halocline layer that's shown here. So ultimately, this layer was enriched in metals, and our question was, is this coming from the North Pacific, or is this being picked up over the continental shelf? We put a lot of thought to that and applied some chemical tracer approaches to sort that question. The first thing that we did was correlate our metals, as an example here, dissolved zinc, with apparent oxygen utilization over the shelf, which is a proxy for biological regeneration. You can see we had a really nice correlation showing that when regeneration was higher at high AOU, zinc was higher. And these were our deepest sites over the continental shelf, whereas surface waters here in purple had much lower concentrations. We also spent some time thinking about the influence of pore waters. And as a tracer of pore water influence, we use the N-star value, which is a proxy for how much nitrate is in abundance or removed compared to its partner macronutrient, phosphate. When you get a negative N-star value, that indicates denitrification has been happening. And we knew that that must have been occurring in sediment pore waters, not in the water column, because water column oxygen concentrations in this area were too high to allow for denitrification. So we have this really nice tracer for pore water influence. And again, we had great correlations between dissolved zinc and N-star, and also dissolved iron and N-star. Again, showing that when N-star was more negative or more denitrification or more pore water influence occurred, the metal concentrations were higher. And this occurred at our deeper depths over the shelf. So all of this confirmed that zinc and iron, manganese are all sourced to that upper halocline layer by regeneration in these Chukchi Shelf pore waters that's then exported out into the Central Arctic via water mass circulation, which is pretty exciting. I also want to talk a little bit about copper and nickel, which I haven't mentioned so far, but have this really unique linear correlation in the Arctic. So here is all of the global copper and nickel data that we have available to us across the global ocean in gray, with the Arctic data shown in color. You can see here in this red, you get a kink in the Pacific in the nickel versus nickel versus copper data. You also get that same kink here in the Pacific, which you can see here in blue. There's no kink here in this Arctic data. We have a nice linear correlation across our profiles. And you can see this played out also in the profiles, characteristic profiles from the Pacific, where nickel's here in orange has a typical nutrient-like profile, whereas copper kind of increases linearly with depth, explaining why you might have a kink when nickel stops decreasing, but copper continues to decrease in the water column everywhere except for the Arctic. So here in the Arctic, the profile shape of nickel and copper is remarkably similar, which is not true anywhere else in the global ocean. And it looks totally different than the rest of the global ocean, with really high concentrations in the surface and really low concentrations at depth. No one would look at this profile and call nickel or copper a micronutrient in the Arctic. So these were really bizarre profile shapes. So we thought a lot about what processes could cause those unique copper and nickel distributions and their similarity. So here are the copper and nickel distributions, again, from the Chukchi Shelf to the North Pole here in the middle, and then back to the shelf here. You can see that the concentrations are very similar for both. They're enriched in the upper halocline layer here in this silicate greater than 25 micromolar contour, and in the surface waters, but surprisingly are not elevated over the Chukchi shelf, which indicated to us that the shelf itself was not likely a major contributing source to the copper and nickel distributions. Instead, you can see based on these plots that nickel and copper correlate extremely well with the fraction of meteoric water, which is water that enters the Earth via rain. We measure this with oxygen isotope measurements and a model that shows that here rain is entering the Arctic via rivers. And so these plots and these beautiful linear correlations are showing that nickel and copper are abundant in the Arctic because of major river sources of those elements. When fraction of meteoric water is high, the nickel and copper concentrations are quite high. And what's neat is over a huge range of geography, we had maintenance of this single linear correlation. So you can see that Arctic rivers are likely a major source of trace metals, at least for copper and nickel, but also for iron. And all of these showed the maxima in their concentrations at the surface near the North Pole, right under the ice, which was there. Surprisingly, I guess, when we got there, there was plenty of ice at the North Pole. Had a lot of problems with ice leading to the North Pole. There wasn't enough for us to sample, which was frightening. So the iron, copper, and nickel and surface waters all have really nice linear correlations with fraction of meteoric water and have their highest concentrations right there at the North Pole. Why the North Pole? You guys might ask. It's because there's an important current that crosses the North Pole. So this is Russia, and over the Russian shelves here, we get river inputs that cross the Arctic really quickly in a current called the transpolar drift that then exits through Baffin Bay and the Fram Strait into the Atlantic here. You can see meteoric water very high in this middle section of the Arctic, exactly where we saw our highest concentrations of iron, copper, and nickel, confirming an Arctic river source. So in conclusion, what I want to say is we had to think pretty hard about what processes are controlling trace metal distributions in the Western Arctic, and especially why those distributions are different than the rest of the global ocean. This is the Geotraces schematic for understanding both the external inputs and outputs, as well as the internal cycling processes that can control elemental distributions across the ocean. And we learned that biological uptake and regeneration don't really drive Arctic nutrient distributions, which is definitely in contrast to the rest of the global ocean, because the productivity under the ice in the Arctic just isn't enough to drive significant in situ cycling. Instead, circulation and water masses really seem to set the subsurface nutrient concentrations, especially in the deep ocean, which I didn't have a chance to talk much about here. And particular to the Arctic is that river and sediment fluxes really dominate the upper ocean metal distributions. In fact, the Arctic could be considered an ideal case study of the influence of rivers and shallow continental margins on metal fluxes to the open ocean, especially in the absence of significant biology, which is sometimes hard to come by. For some future work in the Arctic, we're really interested again in this idea of whether metals are going to play a role in future future primary production limitation scenarios under climate change. And there really have not been very many phytoplankton incubations that study the influence of metals such as iron. So one early experiment that did look at this in 2013 did show iron limitation induced at low light levels. In the Beaufort Gyre, you can see here this white dot is where they added iron compared to their controls without iron, and the add iron grew more. Unfortunately, that's the only place we have incubation. So we've moved to some stoichiometric methods. There was quite a provocative paper published a couple years ago from a European group who studied the Eastern Arctic, again, another Geotraces section, and they were calculating iron limitation based on its relationship to the observed nitrate concentration and the iron to nitrate Redfield ratio, or biological uptake ratio. Beware, this iron star goes negative could be a stoichiometric indication of potential iron stress or iron limitation. And across the Nansen Basin, they found their iron star to go below 0, indicating that iron stress. Now, across the Western Arctic, we use the same values to make calculations, and we didn't see any surface waters that reached that iron stress, iron star. But we're thinking a lot about how that might change in the future, and we really, and also across different phytoplankton groups, actually, we need more incubations understanding the sensitivity of different species to different metal processes. This is my last slide. I think I want to end by saying, we know that increases in ice melt and river discharge are leading to increase in freshwater volume in the Arctic. This has major impacts on both stratification, important for carbon production, as well as nutrient loadings. So thinking about how these new volumes and new hydrologic cycle changes influence the chemical loadings really will require some detailed time series work, both in the rivers and in the coastal areas. And again, we really need more incubations to understand the control of nutrients on different groups in the Arctic. And I thank you very much, and I'll leave it there.
Thank you.